Our next speaker is from the Queensland University of Australia. His name is Harrison Lamb. He has a PhD in animal science. And I saw his speech in January on how he sequenced cattle genome in the fields with the Nanopore technology. And I was quite impressed. Happily, Harry agreed to show today us his research results with the nanopore sequencing in the fields, and we can now see him. Harry, hi.
Hello.
Yeah, perfect. We can see you, we can hear you, and now I can pass the word to you. Can we please put up the slides?
Firstly, thanks very much for having me. It's a good opportunity. And today I'm just going to be talking a little bit about the work that I did during my PhD, which was looking at actually calculating genomic breeding values on farm for Australia's northern beef industry using a new technology called nanopore sequencing. Next slide, please. Whenever we're talking about genetics, what we're really talking about in particular is genotypes. And genotypes are the differences we see between animals. 99% of our DNA and of cattle DNA, they share between each other, but it's really the differences that we're interested in looking at to do our genomic evaluations. Next slide, please. When we're talking about getting genotypes, there's really two options we can use. And Breno touched on this earlier in his presentation, but the first technology is SNP arrays. And SNP arrays have been around for quite a long time, and they're extremely reliable at providing genotypes for your animals. And they provide extremely accurate genotypes, but they will only do genotypes for the specific positions that we have predefined when we load the DNA onto it. Next slide, please. And so I like to refer to SNP arrays as sort of an old Nokia phone where they're great at what they do, which is sending text messages or making a phone call, but that's about as far as they can go. Next slide. The alternative technology we have is called sequencing, and sequencing provides a lot more data and information. And we can get information from whatever DNA we put onto a sequencing machine, whether that be cattle DNA or potentially bacteria or viral DNA. So we get a lot more information from this, but in the past, it has been a lot more expensive. Next slide, please. And so I like to compare sequencing as a technology to iPhones in that not only can they send text messages, make phone calls, but they also offer things like internet, Snapchat, Instagram, and Facebook. So you get many other things out of sequencing than you can do out of SNP arrays. Next slide, please. When it comes to sequencing, there's a few different brands or technologies that you can choose from. And there's one particular technology that we use in our lab, and that's nanopore sequencing. And the reason we use nanopore sequencing is because nanopore sequencing is the only portable DNA sequencing technology available at the moment. And you can see the device there they have is about the size of your hand. So it's extremely portable and it plugs directly into a laptop. So we can basically take that device wherever we want and produce DNA or genotype information wherever we take that device. Next slide, please. So in 2018, actually, Nanopore decided to prove that they could sequence DNA anywhere in the world, and they actually took it up onto the International Space Station to generate some genomic data from bacteria that they found on the International Space Station. Next slide, please. So after seeing that we could take it to places like Antarctica, the International Space Station, and even into the Amazon, we thought in our lab, well, why can't we actually take this device onto farms and provide breeding values or genomic breeding values for farmers on the farms rather than them having to send samples off to the lab? Next slide, please. And the reason we wanted to do this is because Australia has some of the largest cattle farms in the world. One of our cattle farms, for example, is called Anna Creek Station. And Anna Creek Station is about 22,000 square kilometres. And to put that in perspective, next slide, please. That is about the same size as the country of Wales and other countries like Israel. So we have these really, really big cattle properties. And sometimes it can take anywhere from 5 to 6 hours for us to drive from one side of the farm to the other side of the farm. And on these farms, we don't have many fences. Next slide, please. Next slide. And so on these farms, because we don't have any fences, it's really, really difficult for us to actually go out and find all the cows on these farms. And to do that, we use cars, motorbikes, horses, trucks, helicopters, and even aeroplanes to help us go out and find all the cows and then bring all those cows in once a year to sort through them. And that process is extremely expensive. It can cost anywhere from 35,000 to 250,000 US dollars just to do this once. And once we bring those cows in, it isn't physically possible for us at the moment to send samples away to a lab in a major city, which can be a long way away in Australia. Next slide, please. And wait anywhere from 4 to 6 weeks for us to get results to then sort the cows again. Because at that point, the cows are now spread across the farms all over again, and it's too expensive for us to go out and find them. So that's why we want to do genomic prediction on farm. Next slide. Now, I mentioned at the start that there's some other benefits to using sequencing, and those other benefits are that we pick up any DNA that we load onto that sequencing device, not just the cow's DNA. And the reason that's important is because we can do other things with sequencing, like we can detect viruses or bacteria, so we can diagnose disease. We can also look at what bacteria are in their rumens, and then we can predict methane emissions from that. And we can also look at other types of genetic modifications that infer age. So we can actually predict an animal's age using the DNA from a sequencer. Next slide, please. So my PhD looked at basically how we could use this device to go out on farm and produce genomic breeding values for cows. And what we wanted to look at in my PhD was the accuracy of using this device, how long it was going to take, how much it was going to cost, and then what other benefits could we get out of using the sequencing device. Next slide. And so for this particular study or set of results that I'm sort of talking through today, what we were interested in was if we could detect bovine respiratory disease at the same time as calculating breeding values. Bovine respiratory disease is quite a complex disease. And it's caused by a number of different pathogens, but it causes that really snotty and mucousy look you can see in cattle, all that discharge from the nose, and it also causes a fever. And it's quite an issue in large-scale feedlots. Next slide, please. So for this study, we went out and we took nasal swabs from 48 animals in a feedlot that were presenting with bovine respiratory disease symptoms. So they had that snotty nose and they had a temperature and they looked like they were sick. And then our goal was to actually generate breeding values and also diagnose whether they had bovine respiratory disease or not within 24 hours. Next slide. For our genomic breeding values, we wanted to check that we were actually delivering accurate breeding values. So we also sent some DNA off to the labs to get our official Australian breeding values for these animals, and then compared our Australian breeding values to the breeding values we could generate on farm. Next slide, please. For the pathogen detection, again, we wanted to check that we weren't just picking up any bacteria or viruses for these samples. So we also sent the bacteria and viral DNA away to a lab to double check what results we were getting from the lab, and then compared those results to the results that we got on farm. Next slide. Here are the results for our accuracies. You can see on the left there, we have our SNP array accuracies, and these are the accuracies of genotyping 700,000 different positions across the animal's genome. And you can see in Australia, our SNP arrays for commercial producers are roughly 88% accurate. And the sequencing data we got on farm, we got a genotype accuracy of around about 89, 90%. So we actually got better accuracies than we did for the SNP array. Next slide, please. Here you can see the correlations we got between the two different technologies using the SNP array and using our sequencing technology. And these are for 4 different traits that we calculated on farm. And the correlations here you can see are the correlations between what we calculated either from the sequencing or the SNP array from the lab and to actual or essentially true breeding values that we had for these animals. And you can see the correlations there were quite high for the 4 different traits that we had, which were body condition score, body weight, heifer puberty score, and hip height. Next slide, please. Next, we looked at the bovine respiratory diagnosis. Next slide. And we actually got a 95% match between the different bovine respiratory disease pathogens that we picked up using our method and the pathogens that we detected when we sent the samples away to the lab. In this, we also included 2 negative control samples, and these were tail hair samples. And we wanted to include these because we wanted to check that we weren't just picking up any random viruses or bacteria, and that we were in fact not going to give back false positives. And for neither of these 2 samples did we detect any bovine respiratory disease related pathogens. Next slide. Next slide again. So here you can see a heat map, and this sort of just displays the different species or bacterial and viral pathogens that we picked up when we were on farm. Next slide. And what you can see is that on the x-axis, we detected a different species that was related to bovine respiratory disease for almost every single sample, except for the 2 negative control samples there, where we only detected bacteria that were found in dirt, as you could imagine would be present on a tail hair sample. Next slide, please. I mentioned cost. Cost was another key driver for this particular study because we wanted to check that it was in fact going to be cost effective for producers in Australia. Next slide, please. In the current study that we did, we tallied all the different costs that we had for just generating the data and the breeding values. And that came out to be about $24 per sample, but this doesn't include labor. And it doesn't include any of that overheads like driving out onto the farms and taking samples. Theoretically, we think we could increase the size of the run up to 72 animals, and this would decrease the price down to about $16 per sample. And in Australia, our SNP array genotyping that is commercially done for farmers, we get that for around about $34 per sample, but that does include labor and it does include overheads. So you can see roughly how they compare, but again, that's not a like-for-like comparison. Next slide, please. So where are we up to? Next slide. We know the accuracy of the technology, and we know that it's quite accurate. We know that we can also detect pathogens at the same time from the same sample. So this means we could potentially detect things like pestivirus while we're genotyping, or other diseases that might be prevalent in a particular region. And we can do all of this in 24-hour turnaround for about 50 samples. And at the moment, this together costs about $25 per sample, excluding labor. Next slide, please. The problem we still have, though, is that it requires a lot of equipment. So here's a snapshot from another researcher of some of the equipment that we need to take out onto farm just to do all this genotyping. And there's simply just too much equipment at the moment, particularly when you're dealing with cattle and you're trying to do things quickly. It just becomes a little bit overwhelming. So next slide. One of the things we're looking at there is actually to automate some of these procedures. Next slide, please. So we're looking for automation, like an automatic robot to do a lot of the sample prep, and this would potentially increase our throughput up to about 200 animals per day. And we're also looking to optimize the methods further to decrease the price even more. Next slide, please. So that was where I got to in my PhD, and we're still obviously working to make this a reality on farm. I need to briefly acknowledge my supervisors for their help on this work, Meat and Livestock Australia, and some of the collaborators in the Northern Genomics Project, as their data was really crucial to this, as well as all the different members of our lab back home in Brisbane. Next slide. And so with that, I'd just like to open the floor for any questions. Thank you.
Thank you, Harry. That was quite interesting. Let me check for the questions. Any questions from the audience here? Okay, and from the translation, I can see any questions as well so far. Okay, thank you anyway. If any questions comes up, we will send them to you for you to answer them. Thank you very much. That was quite interesting.